Hi everyone. In this video, we will focus on graph-based clustering approach that Seurat takes in single cell data analysis to cluster your cells into different clusters. Okay. So the typical single cell data analysis steps. So these are the things that is followed. Like that means you take the raw data, you do the filtering, then you do normalization, scaling, find the most variable genes, and you take that, and then you do the PCA analysis. So PCA will help us to get the signal and remove the noise and compress the data from, let's say, total 2000 genes to 50 metagenes. So in the last video we talked about PCA and how PCA helped to remove the noise and get the signal to obtain the 50 metagenes, using which we can do the downstream analysis. So now PCA space what we got, or PCA coordinates what we got, that forms the basis of the single cell downstream analysis, and the next step is the clustering. And after clustering, we do the visualization using UMAP or t-SNE based approach, and then we do the differential expression, and then we do the cell type annotation. So in this video, we will focus on the clustering, because this is the important step, because how clustering is actually calculated and how tuning the parameter will change your clustering, and all the downstream analysis will based on this clustering. So that's why we will focus clustering in this approach, okay, in this session. Okay, so basically Seurat does these many steps to come to the clustering. Okay, so first initially, before this step, we obtain the PCA matrix. That means we don't have now genes. So now we are playing with the meta genes or the principal components, where each row is equal to, let's call, one principal component or one meta gene that contains information from all the genes, and each column are the cell. So we got now PC cross cell. So let's call this a PCA matrix. So this is what Seurat first uses, and then in this PCA space, it now tries to calculate the distance between each cell. So we will get a cell to cell distance matrix based on the PC coordinates. So now this gives us information which cell to every cell, how the distance is, whether those are close in terms of their expression profile or they are very farther apart. So next what it does a k nearest neighbor graph based approach. Okay, it does k nearest neighbor for where every node is equal to one cell, and if there is a connection between those two nodes, that means this cell and this cell are in KNN space or k nearest neighbor space, their expression patterns are very similar. Okay, and this is called the KNN. And after that, it then something does called SNN, that means similarity based network graph. It does SNN, and I will talk about that. And once that SNN graph is done, then it does the community detection. So we'll talk about each step one by one. Okay, so that's what is very important. In the previous step, we performed PCA to reduce the dimensionality, and now we use those principal components to identify groups of similar cells. And that's why I have explicitly written that clustering happens in PCA space, not the original data. And also it doesn't happen in UMAP and t-SNE. Okay, UMAP and t-SNE are the for visualization. Okay, PCA is must. So without PCA, you cannot do all that. So some people get confused. What is PCA, t-SNE, and cluster, UMAP? But as again, foundation, like PCA is must. So PCA, based on the PCA space, you do the clustering, and then you go for UMAP and t-SNE for the visualization. So basically, clustering, what it will tell, cells with the similar gene expression profile will go into one clusters. Okay, so that's what we will see. So first, what it does is k nearest neighbor graph. Okay, so that means for every cell, what you need to do, you need to find let's say k most similar cells. Okay, you do a boundary in the nearest neighbor space and find the, let's say, top 10 similar cells. Okay, and for example, you calculate cell A, which are the neighbors, and cell B, which are the neighbors, and that way you create a graph. So where each node is equal to one cell, and if there is a connectivity, they are neighbors to each other. So that way you do a k nearest based graph. Okay, so that's why cells that are close in PCA space will become connected in this KNN graph. Okay, but this is still not enough. That's where it does another improvement, that's called shared nearest neighbor graph or SNN graph structure. So here the logic is like two cells are strongly correlated if they share same neighbors. Okay, for example, let's say cell A, the neighbor of cell A is BCD, and E has also BCF. That means these A and E share two close neighbors, B and C. That means their weight is now 2. That means more you and me have similar friend circle. That means you and I are very close. So that logic, so with that logic, again you are giving weightage to the connection. So KNN will just tell us who is connected to what, but SNN will tell us how strongly they are linked to each other. So it will give more weightage to the linkage. So now we get a SNN graph, and now we can do something called community detection algorithm. So basically the algorithm, there are different ways of clustering, like Seurat uses algorithm like Louvain and Leiden. So the logic is like you have to break, or uh, the network such that within distance is very high, whereas between distance is very low. So that means within a cluster they are more closely related than between the cluster. For example, this blue is more connected to blue compared to blue is connected to red. So that logic, so with that logic we will clustering that. And while community detection, there is a function called FindClusters method. There is something called resolution is a very important parameter. So if you are doing resolution very low, so that means you do not want to do fine grain. So basically you are interested in more superficial, that's why resolution is low resolution, and that's why you will get few clusters. But if you want to do very deep clustering, fine grain, so then you have to increase your resolution parameters, and that's what for example, if you go to let's say 1.2, you will get more clusters. So higher is the resolution value, more granular will be your cell population. Okay. So if you come back to Seurat tutorial, so that's what they have mentioned here, like they're using a graph based clustering approach, and initially they calculate the distance matrix, and again that matrix is derived from the PC space, not the original space. And then what they have they are saying, like recently they are using what is this KNN graph based approach, where with edges drawn between cells with similar feature expression pattern, and then they attempt to partition this graph into quasi or the communities. Okay, but recently as per Finograph, so now what they do, they are constructing a KNN graph, and then they try to find neighbors, this find neighbors function, and based on that they try to find the local neighborhood or who is connected to what. Okay, for the KNN they use this find neighbors method, will help us to do the KNN who neighbors, it's identification of neighbors. But find neighbors is not yet clustering, so this will just tell who is connected to what. Okay, we have not broken down the clusters yet. So for the clustering we have to use the next function called find cluster. So find neighbors function will tell who is connected to what. We'll get a graph, but we'll get two kind of graph, one is KNN graph and one is SNN graph, and next step basically use this graph to break that graph into different cluster. So that's what they have also mentioned here. I was talking about the SNN, right? Yeah, this is the SNN click method, right? So they are using that as well, SNN graph. Where is the written? Okay, so what they have telling, they are using the Louvain algorithm or SLM based, to group the cells, and main what is the goal of optimizing the standard modularity function. So that means module, each cluster is one module. So the logic, like within the module they are connected strongly, and across between the cluster they are not connected like that. Okay, again as I said, granularity will tell us how many clusters you want. Okay, so these are the two steps what we will do. Okay, so let's okay, so we have done the PC analysis, right? So that's what we have got PVMC at the rate we got reductions, so that will give us the PC. Okay, so we have 50 principal component with the data, and total number of cells are 2638. So our dimension is now 2638 cross 50. Okay, so now we will do the first thing, like we are doing the neighbors, right? That means KNN and SNN graph. So if you before that, if you go PBMC object, there is something called graphs. The slot will be there, which will be empty because we don't have graphs yet. Okay, so now when you do this find neighbors function, okay, so let's check this find neighbor function, what are other parameters are there. Okay, So other parameters are there, but I will let's go over here. So default, you say that you, instead of 50 principal component, just use 10 principal component, right? Because that will be more faster, and we have seen that 10 is enough. So here you can see, you calculated 50 principal component, okay, PC, but to use, you can choose how many principal component you want to use for the clustering, because more information you add, more noise you are actually adding. Okay, so here let's go with 10 because that's what we see here, and we do the neighbor. So it will first calculate the KNN based graph and then the SNN. So now if I again check this graph, right, pbmc.graph, I will see that we'll get a graph object containing 2638 cells, and SNN is 2638 cells, but still we don't have clusters. Okay, so now if you just do pbmc at the rate, I think yeah, so clustering will come here in the metadata. Okay, which cell belongs to which cluster. So let's do one thing. Okay, first I will show you head of this one. You see that we have only this much metadata. That means these are the cells, each row barcode, and the original identity is like the number of RNA, number of genes, percentage of mitochondria, and we have categorized it into high mitochondria or low mitochondria class, and you see no more information. So now when I do the clustering, right, so basically it will now do the clustering, and it will put each of these cell into a particular cluster. So we are going with this resolution 0.5. Okay, let's see default one. So what it did, like, it's the graph was around, see, it started with the graph, and there were 2638 cells were there, and number of edges were around 95,000. You see, that means how many connected uh, they're very highly connected, edges are there. And then it uses the Louvain algorithm, and with this resolution it gave us total 9 community, means we got around 9 clusters. Okay, and if you want to see now head metadata, you will see that we have got now two extra this thing. One is the Seurat clusters and the RNA SNN based clustering it used. So mostly they will be very similar, but this is the parameter that tells like which barcode goes to which cell. Okay, yeah, and if I go to pbmc at the rate reductions, you will see only the PCA, right? So, and just to see, I wanted to see like whether UMAP has come or not, clustering has got generated or not, but because we have not done the UMAP, so that's why we won't see. So okay, so we got this PBMC metadata, and that tells like which clusters belong to what, and then just to see which 5, so you can see that this barcode is belongs to second cluster, this barcode becomes third cluster, like that. Okay, and also you can do a summary of PBMC at the rate metadata dollar let's say Seurat cluster. It will tell that there are 684 cells belonging to cluster number 0, 481 cells belonging to the first cluster, and like that. So that means these are the 18, and those are the very rare clusters. I mean, number of cells are very less. So you can also identify how many clusters got into each, how many cells got into each clusters. Okay, and yeah, so as I said, like find neighbors method, right, you can choose how many neighbors you want. I think that is around I think k neighbor parameter, like 20 neighbors you want for every cells to construct the graph. And yeah, and the other thing is the find clusters. So here the most important parameter is the resolution. Okay, so by default one is 0.8, and I used 0.5. So as I said, if you increase the resolution, you will get more this thing. Okay, so let's try to see again rerun. Let's say 0.8. Okay, and it will redo, and let's do, so we got 11 clusters, you see. Okay, so you can also, I'm just adding this one here as well, summary. So you will now see that initially it was these numbers are very much similar, little bit reduced, see, 542 and 428 here. So I think between 0 and 1 cluster, some part moved into other cluster, but remaining Also low also I can see. Yeah, that's what very different clusters you will get when you move to different this thing. Okay, resolution. Okay, so as per the documentation it says this one, and then after that what we need to do is to visualize it. So that's where what we need to do is like either we can go with UMAP or t-SNE based non-linear dimension reduction method. So basically what it does, it takes the now we got the PCA space, right? It further applies non-linear information to visualize. Okay, just to show you one quick visualization. Okay, so you can just, you will see this kind of plot. Okay, so now in this example with the default we got 10 coordinates, right? 10 clusters, and so the it will be, this is one cluster, this is one cluster, like that you may plot we will get. Okay, so yeah, I think what if I just summarize what we did, like in the space, we cluster the cell and we talked about the different algorithms, how it works and how to tune the different parameters. And yeah, so now we need to visualize. So that's where in the next video I will focus on the visualization part, and we will talk a little bit about PCA versus UMAP versus t-SNE, and when to use what and how the visualization will change. We'll see that. Okay, I hope this was useful, and thanks for watching. I'll see you in the next video.